Good morning everyone. In the previous class we have learned about the cell division and continuation of life. Okay, so let me learn about the scheme of the mitotic division. Okay, the scheme of mitotic division was confirmed in 1904 by Theodore Boveri in 1862 to 1915. The chemical nature of the genetic material was determined as a series of experiments over the next 50 years. Okay, so as we see it in the table here regarding cell division on the figure 27, as we see that the egg cell is of a haploid nucleus and the sperm cell is having haploid nucleus. These two haploid nucleus together forming diploid nucleus. That means they are forming 2n fertilized egg. Okay, this 2n fertilized egg undergoes the cell division. Again, it will split into two nucleus with the same number of chromosomes. Okay, so same number of nucleus. This 2n nucleus is dividing into two cells, and these two cells are having again 2n nucleus. Okay, these two cells will split into four cells. That means one cell is giving rise to the two cells. Okay, these two cells are having again the same number of nucleus. Okay, these four cells will split into 8 cells. Okay, one cell that is from the posterior side, the nucleus is transferring. So the nucleus is having either the sperm cell or the egg cell. That means one cell is splitting into n nucleus. That means one cell is dividing into the haploid nucleus. That means it may contain either the sperm cell or egg cell. Okay, series of experiments over the next 50 years culminating in the determination of its structure. The deoxyribonucleic acid in 1953 by James Watson and Francis Crick, scientists proved that mitosis takes place in all the body cells which retain same number of chromosomes. So James Watson and Francis Crick has observed the DNA structure in 1953, and scientists proved that mitosis takes place in all the body cells which retain same number of chromosomes. That means while the division of the body cells, but the chromosome number will be the same. Okay, meiotic division takes place in sex cells where the chromosome number is halved. Okay, as I told before, that is one cell is splitting into haploid nucleus. That means one cell is dividing either into either the sperm cell or the egg cell. That means the cells will always contain only one set of nucleus. Okay, next about cell division in human beings. Okay, so we have studied about the single celled organisms and the plant cells and animal cells. That means eukaryotic organisms. Let me see the cell division in human beings. Okay, we know that cell as the structural and functional unit of life of any organism. In all organisms, the cell divides and forms new cells. The process of the cell division is same in unicellular organisms and highly evolved multicellular organisms like human beings. As we know that cell is the structural and functional unit of the life of any organism, either they are single celled organisms or multicellular organisms, the cell division is same. Okay, and also in all organisms the cell divides to form the new cells. Okay, as Virchow stated that the new cells will arise from the already existing cells. Okay, cell division is the process that transfers a human fertilized egg into a baby in 9 months and into an adult in the next 20 years. Cell division and function in a multicellular organism is highly regulated. It occurs only when there is a need for it. Okay, cell division is the process that transforms a human fertilized egg into a baby in 9 months and into an adult in the next 20 years. Okay, as we know the cycle of a human beings, that means when the egg cell is fertilized, so after the cell division of 9 months, this organism, so the individual is completely formed, and these individual which was formed after the division of the cells on 9 months, so it will transfer into a complete individual, and after 20 years it will transform into the adult. Okay, cell division and function in a multicellular organism is highly regulated. So as we know that in the multicellular organisms, the division of the cell division is highly regulated, and it occurs when there is need for it. So as we see that in the 20 years of the lifespan of an human beings, we will see different stages. Okay, that means in the different stages, different cells will divide, and the cells will also originate to form the tissues, and those tissues will originate to form the organs, and those organs will form the organ system, and these organ system will help to grow the individual according to the mass and its height. Okay, cells in some organs such as heart and brain of an individual never divide. Okay, so we have to see this point carefully, that is, cells in some organs such as heart and brain of an individual never divide. Okay, on the other hand, bone marrow cells actively divide to produce red blood cells which have a short lifespan of 120 days in the body. For example, if you cut your finger and bleed, so a blood clot forms to stop the bleeding. This brings in various chemicals to the site that stimulate skin cells to divide and heal the wound. Cell division ceases as the wound is completely healed. In contrast, cancer cells do not respond to such growth regulating factors and continuously divide at the expense of the normal cells, thus ultimately killing the host. So it becomes important to understand the process involved in cell division. The cell cycle will help us understand this better. Okay, so as we see in the bone marrow, it always divides the red blood cells. That means after 120 days of a lifespan of the RBC, the RBC will destroy, and the new RBC cells, that means new cells are arising from the already existing cells. Okay, and also, for example, if for suppose we have cutted our finger, okay, what happens? It starts bleeding. For a course of time, what will happen? It again starts to form clotting. Okay, after a certain days, what will happen? The scare which was happened due to the cutting of our finger will be disappear. That means the cells are completely forming to produce tissues. Okay, those tissues will make us free from that scare. Okay, and another example regarding the cancer cells may not destroy, but it will completely destroy the host. That means the cells will completely divides continuously and they will kill the host. Okay, so it becomes important to understand involving the cell division. Okay, here as we seen in the red blood cells, what will happen? The previous cells may destroy and the new cells are producing. So in the cancer cells, what will happening here? The cells will continuously starts growing, but they are not destroying. They will destroy and kill the host. Okay, next, to understand this better, let me know about the cell cycle. The process of cell division is called mitosis. Okay, so the process of the cell division is called mitosis, which is completed in 40 to 60 minutes. This is the time to active division. So this mitosis will complete in 40 to 60 minutes. Okay, the period between two cell division is called interphase. The period between the two cells, that means as we learned before that 2n nucleus is dividing into equal sets of cells. That means the single cell is giving rise to the two cells. The two cells will have 2n nucleus. Okay, this is actually the period when the genetic material makes its copy so that it is equally distributed to the daughter cells during mitosis. That means the cell is giving rise to two daughter cells, and the two daughter cells will resemble the parent by the equal set of chromosomes. Okay, Interphase can be divided into three phases. Here, the interphase will be divided into three phases. First one is Z phase, S phase, Z2 phase, and M phase. Okay, let me know about the Z phase. This is the linking period between the completion of mitosis and the beginning of DNA replication. The cell size increases during this period. So, in the Z phase, what is happening here was the DNA starts replication. Okay, the cell size increases during this period. Next, S phase. This period of DNA synthesis leading duplication of chromosomes. In the second stage, what is happening here? The DNA starts synthesizing, it produces, so it will leading to the duplication of chromosomes. Okay, in the Z2 phase, this is the time between the end of the DNA replication and the beginning of mitosis. Cell organelles divide and prepare chromosomes for mitosis. Okay, in the Z1 phase, what is happening here? The DNA starts replication and the size of the cell increases. In the S phase, what was happening? The DNA starts synthesizing and it is trying to produce chromosomes. Okay, in the Z2 phase, this is the time between the end of the DNA replication. So, in the G1 phase, okay, DNA replication starts and increases the cell. In the G2 phase, the DNA completes its replication, okay, and the beginning of mitosis. Cell organelles divide and prepare chromosomes for mitosis. The cell organelles will divide and they will prepare the chromosomes. Okay, in the M phase, this is mitotic cell division phase. It includes prophase, metaphase, anaphase, telophase, and cytokinins. So, in the M phase, the mitotic cell division takes place. That means it includes prophase, interphase, metaphase, and anaphase, telophase, and cytokinins. Okay, to understand the functional relationship between these phases, Portu Narasimha and Johnson conducted some experiments using the cell fusion technique. This is combining two cells in experimental conditions. With the cell fusion technique, Johnson and Portu revealed for the first time the structure of the interface Z1 S phase and Z2 phase chromosomes that are not ordinarily visible under the microscope. They provided evidence on progression of cells through the cell cycle in sequentially unidirectional and controlled way by a series of chemical signals that can diffuse freely between nucleus and cytoplasm. These experiments are considered to be a milestone in the cell cycle studies. Okay, so later on, Johnson and Portu Narasimha Rao, they has studied about the functional relationship between these phases. Okay, as we studied before that, one nucleus from the sperm cell and one nucleus from the egg cell are two are fusing together and they are producing a daughter nucleus. That means it is having two nucleus. Okay, again this parent cell is giving rise to two daughter cells. Okay, the two daughter cells will produce same number of chromosomes as the parent. Okay, the two daughter cells which are formed in the stage 1 will split again into 4 daughter cells. Okay, these two cells are giving rise to one cell, these cells individually giving rise to two cells. Okay, again that cell will individually gives arising to a nucleus. That means it is also producing the same number of cells as it is divided in the stage 1, and these cells will have equal number of chromosomes. Okay, they provide evidence on progression of cells through the cell cycle in sequentially unidirectional and controlled way by a series of chemical signals that can diffuse freely between nucleus and cytoplasm. Okay, they has observed about the chemical signals that are diffusing freely between the nucleus and cytoplasm. Okay, so these experiments are considered to be a milestone in the cell cycle studies. Okay, it is concluded as a milestone in the cell cycle studies. Okay, so with this, cell cycle has completed. In the next class, let we see about the different stages of mitotic cell division. Okay.